Pleasure to introduce Laura Silverman today as our keynote speaker. Laura earned her degree in chemical engineering from Princeton and a doctorate in bioengineering from Penn. After 7 years as an assistant professor in neurosurgery at the University of Tennessee, Laura joined a startup focused on developing tissue engineering products for spinal discs, where she served as their head scientist for the past 10 years. Laura is particularly interested in innovation at the interface between academia and industry and is currently serving as the chair of the Industry Affairs Committee for the Orthopedic Research Society. We've asked Laura today to speak to the challenges in bringing new tissue regeneration technology to human clinical trials, particularly from the perspective of a female scientist. She's been in the trenches and has the arrows in her back to show for it, and I think speaks from a viewpoint that's particularly relevant to our stage 3 ITP. So thank you, Laura, for making time, and please go ahead and share your screen.
All right, thank you so much. And thanks for the invitation to participate today. It's been wonderful seeing a lot of these presentations and seeing how far along a lot of these products are in their development. So today what I want to do is give a little bit of background on cell therapy, because that's my particular area of expertise, and talk about some of the areas that I think are important to focus on as we translate from those early stage ideas into that clinical trial, those early clinical trials, because that's a really critical stage of development. And there's a lot of big lessons that need to be learned in order to successfully cross that gap. And then we'll talk a little bit about academia versus industry and some of the things I've experienced over the years that may be of interest to you all. So regenerative medicine seeks to augment, repair, replace, or regenerate various areas of the body. And a lot of the presentations over the past 2 days have covered such products. The Alliance for Regenerative Medicine is an industry organization that tabulates a lot of statistics around this field, one of which is the tremendous amount of funding that's found in the cell and gene therapy space. My specific expertise is in cell therapy, where we use the actual cell as the therapeutic component. And it's ideal for tackling previously intractable diseases where traditional therapeutics just haven't been efficacious. The reason is because these cell therapies often present novel and often multimodal mechanisms of action. So I've worked on a product where not only did cells regenerate tissue, but they also released anti-inflammatory cytokines. And especially in complex diseases where perhaps there are multiple etiologies, multiple modes of action, can be highly efficacious in the clinic. And cell therapy is a really exciting space because there's radically new science that's come to fruition here with the advent of CAR-T, with induced pluripotent stem cells, CRISPR gene editing. The gene-edited cell therapy space is absolutely booming right now. Cell therapies are regulated by the FDA. We've had a lot of discussions today about the FDA, specifically the CBER department and OTAT. And cell therapies are interesting because they often are presented as combination products, especially with medical devices, which means you also have the CDRH department involvement. And this interplay can certainly pose some interesting challenges. So there's been a lot of news coverage of the regenerative medicine space. I'm not sure if you guys saw, just about 2 weeks ago, there was a beautiful article in the New York Times describing some groundbreaking data that was generated to tackle type 1 diabetes. And the treatment is based on the delivery of embryonic cell-derived islet cells, insulin-producing cells, and they were delivered into the first patient and they saw remarkable results. So there's a lot of interest in promoting these products, supporting these products, but certainly limitations still exist. This particular product developed by Vertex, which was acquired from Sema Therapeutic, requires constant immunosuppression. And so there's definitely room for improvement for these patients, because they'd have to exchange having diabetes with being on long-term immunosuppression. There's also, in this particular case, there's been a lot of discussion in the news around the cost of insulin. And so having a single treatment would certainly be better than the long-term costs of insulin. Now, cell therapy can be broken up into a number of categories. One type of cell therapy is autologous cell therapy, meaning you take cells and tissues from a person, send it to a manufacturing facility and send it back to the person. Another model is allogeneic cell therapy, meaning you take cells and tissues from a person, you manufacture them, and then you can give them to many people. And there's a lot of economies of scale with this model, but sometimes you can hit immunological issues such as rejection. Another way to characterize cell therapies is to divide them between 351 and 361 products. So the 351 products are considered manipulated, which means there's extensive oversight by the FDA of these products. So anything that's grown in a flask, anything that's gene edited is manipulated. The other category is called the 361 category, and you would self-designate yourself as being what's termed minimally manipulated, which means you're not actually changing, these cells or tissues. And that results in no requirements for clinical trials or potency testing, which is limited oversight. Unfortunately, what happened was a number of groups were self-designating as 361, and patients were finding themselves in situations where their product was not manufactured appropriately, and they were either having contaminations or they were actually getting hurt by the product. So FDA decided to set up some programs to try to transition all these companies into the 351. And they set up a discretionary enforcement period where they said, okay, guys, time to switch over and start following the 351s, submit your IND. The discretionary period was supposed to end in 2020. With COVID, it got pushed back. So this year, they actually ended the discretionary enforcement period, and a number of companies have shifted over. And that's actually, especially in the orthopedic space, that's actually caused quite a shift in the competitive landscape as these companies have had to change their regulatory pathway. So there are a number of approved cell and gene therapies. Half of them are in the immuno-oncology space, the first one being approved in 2010. 2017 was a really exciting year with the approval of Kymriah and Yescarta CAR T therapies. And very exciting, this year there have been 3 additional immuno-oncology products that have been approved for use in the United States. All of these are autologous, as you'll see, so there are a number of companies chasing allogeneic CAR T, and it'll be interesting to see what happens to these very expensive autologous products when those allogeneic products become available on the market. Oops. There are a number of other approved cell therapies on the market, including one in the dental space, Gentuit. One product of note here is Zolgensma, which also got a lot of press attention. This was approved in 2019, and the attention it got was not necessarily positive. It is one of the most expensive treatments to ever be sold, with a price tag of over $2 million per treatment. There have also been an additional 2 products that have been approved this year for a total of 5 in 2021. And that's really remarkable considering the backlog and burden that the FDA has been experiencing with COVID. It's really signaling a return to business as usual and a willingness to start focusing on non-COVID products and indications again. Of course, there's a lot of activity outside of the US. Certainly they're the most therapy developers here in the United States. There's also a lot of them in the Asia Pacific region. Japan has a specialized regulatory pathway. South Korea was an early adopter of cell therapy and regenerative medicine. There's a lot of activity in Europe as well, but interestingly there, the reimbursement landscape is quite fragmented. So even if you can get approval, it's quite burdensome, and quite a lot of resources are needed to go country by country and region by region to secure reimbursement. So that becomes a little bit more challenging as you put together an entire market strategy for a new product. So at the core, we're all here because we've identified some excellent new science. And as I've been working outside of a sponsor, now I'm a consultant and I work with a lot of different small cell therapy companies to help them translate their products into the clinic. I think one of the common threads that I see is a need to understand that excellent science is not enough. Even in this stage of development, you're still many years from a BLA. Even in the early stages of development, science really is not enough. And there's a lot of different factors that even spending a little bit of time on now will really help you as you think about interacting with the FDA and also raising funds outside of grants and with the wider market. So I wanted to touch on some of those today. So the first thing to know is that you are not alone. You need to understand the competitive landscape for the indication. And this is divided into products that are already on the market that may have a totally different mode of action or categorization, and also products that are in development. For each of these products, you need to understand the company that makes them, what's the product and how does it work? How does it compare to your technology, and what stage of development or how much does it sell for? And finally, what is their preclinical and clinical data if it's relevant? And the reason is because this information is critical for a number of reasons. One is it serves as a regulatory pathway cheat sheet, especially for the preclinical and clinical testing. For better or for worse, when you go talk to the FDA, they'll likely be looking at precedent products in the same space, and you'll be expected to meet a lot of the same bars as the companies that came before you. I've experienced that firsthand. Sometimes the studies don't even make sense, but because other companies did them, they want you to do them as well. There are also this kind of information is also really critical as you have discussions with investors. And importantly, it can guide messaging to ensure differentiation from your competitors. When you're early in development, you still have some flexibility. You can still pivot, you can still change features. 3 or 4 years down the road, or when you're in clinical testing, it's much harder to make changes, and you don't want to find yourself being a Me Too product to something else that either is on the market or about to come onto the market. Now, another question to ask yourself is, do the numbers work out in your favor? So you can work backwards, looking at the cost of the current treatments or care, looking at the cost of overall burden of disease and the existing reimbursement codes, basically to figure out what you can charge for your idea. But looking at it from the other direction, working forwards, you can understand how much it will cost to buy your raw materials, how much will it cost to make this product. There's a baseline cost just to use a clean room. And of course, you can apply technology to drive down manufacturing costs, but there's a lot of upfront time and money spent in these kinds of efforts. And then also, what is the cost to lot release your product? In the cell therapy space, lot release testing can be tens of thousands of dollars, up to hundreds of thousands of dollars if you have bovine components in your raw materials. So it's really important to understand what are your costs and if you'll be able to recoup those when you actually perform sales. A number of you have spoken to patents. I'm very happy to see that, because not all groups are as aware of this. You need to have a good patent portfolio. You need to have patents that show that your process and your product and your tests are novel, useful, and non-obvious. And this serves for defense, to keep your product uniquely yours, but also as offense, to keep others out of your space. If you can patent permutations of what you do, it'll keep people away from you. And you need to decide what markets to enter with these patents based on where you intend to sell the product. And this is really, really important as you start to fundraise, because it informs the long-term business opportunity to your potential investors. Now, we all know that the FDA is the gatekeeper, and their job is to protect the patient. This may be controversial, but I will say that FDA is busy, the FDA is tired, and we're expecting the FDA to understand complex and novel science very, very quickly. And so I spend a lot of time working with my clients to help them to keep it simple in these interactions. You want to use a very clean target product profile. You want to use non-technical language, which can be difficult for those that are used to being in academia and writing grants and manuscripts. The more you can simplify your language, the better. The use of diagrams and images is encouraged, and of course, document formatting is really important. It sounds trivial, but it makes it easy for these reviewers to quickly move through the information that you're presenting to them. Oops. Sorry. And I think one other note here is that the FDA will not provide endless feedback. This has happened to a number of my clients where they kind of used up their card with the FDA. So it's really important that when you have a touch point with the FDA, that you ask really meaty questions that impact your development pathway. And they're not questions that you can find answers to in guidance documents, because at some point the FDA will say, you're done, you've asked all your questions, submit the IND, good luck. And it's difficult to be in that situation if you still have actual questions for them. Another consideration early on is that quality is king. Even during preclinical development, you need to consider quality from day one. Quality represents the checks and balances within a company, but even at the simplest level, it enables document storage and data integrity. So if you're having an FDA discussion or an investor discussion and they say, oh, I want to see the report for animal study 6, you need to be able to pull that quickly, and you want to make sure you have all the data together. Another important aspect of quality is that a lot of development is based on risk management. You can't test everything. You have to sometimes, pick the things that have the highest risk. And that's an activity that's often led by quality. So some of the initiatives you want to consider even early on are having a basic quality manual, having quality assurance, and certainly having a document storage system. It's something that early stage companies sometimes overlook. Now, the GMP manufacturing is certainly a very long process. In cell therapy, it can be quite a journey to get from your research process over to an optimized cGMP process. Often you'll enter the clean rooms with a more basic process and optimize as you go, although sometimes it makes more sense to optimize right out of the gate so that you don't have comparability challenges. As you enter the clean room initially, you need to think about assay development. You need to remove subjectivity and variability in your process. There's a lot of documentation to establish, probably a lot more than you realize, and you need to consider updating your raw materials, especially to remove proprietary raw materials where you don't know the composition, and also trying to minimize materials that contain animal components or human components. As you think about getting closer to your BLA and having a process that you perform 24 hours a day, 7 days a week in a commercial context, this is where you want to start really thinking about applying technology. How can you, reduce the number of staff that you have in there and really make this a routine production? There's going to be a lot of collection of data, analysis of data, trending, and the use of quality by design is really valuable here as a tool to document all of this development and continual improvement over time. So here's just a very simple example in cell therapy of the kind of development that we have to do. So originally you can grow cells in a single flask, but as you want to scale up, maybe you'll start using multiple flasks that you stack on top of each other. At some point this becomes cumbersome, so you can use a multi-layer flask, which is actually a flask that has one singular port and you can fill with media. You tip it over and the media equilibrates across all the levels, and then you tip it back, and now you've been able to do a media exchange on a much larger surface area with just one intervention and one person in one event. And this is something that you can certainly do. I have a client who does this, this brute force method. There are some pros to this approach. Alternatively, you can transition to something like a bioreactor. If your cells grow in suspension, that's easy. If they don't grow in suspension, you can use something like a microcarrier. You can see here, the cells actually will proliferate on the surface of the microcarriers, and the microcarriers will float in the bioreactor. You become quite reliant on the manufacturer of that bioreactor, but it does enable scale up much easier than trying to just have more and more multi-layer flasks. So ultimately, the goal of manufacturing is to consistently produce a safe and efficacious dose. And cell therapy specifically, we look at 4 things. We look at safety, identity, purity, and potency. The potency assay development should begin very early based on the modes of action you identify for your product. And then over time, once you understand more and more of your product, you can start to refine your assays and remove irrelevant assays until you're finally ready to validate them and go to a BLA. Now, the animal study program is one that garners a lot of attention and discussion, obviously, because it's lengthy and expensive. Certainly in cell therapy, there's a high burden of requirement for the animal testing. And I have a number of clients that will say, oh, well, I published a lot of papers with animal models. Are we good to go? And unfortunately, the answer to that is no. And the way that we know that is through the preclinical assessment of investigational cell and gene therapy products guidance documents. I've heard a number of you all referring to these guidance documents. They're very critical, and they help us understand what is needed. So in the cell therapy space, they articulate 2 phases of animal studies. The first is the proof of concept phase. And there's actually quite a lot of different studies that need to happen here, not just 1 or 2, but maybe 6 or 12. You need to establish the injury model, look at survival and engraftment, distribution, differentiation, tumorigenicity, et cetera. When you transition to phase 2, really it's only 1 or 2 studies, but these studies typically cost 2 to 3 times as much. And the reason has to do with specifically compliance. So during the early phase, there's not a whole lot that you need to do besides some basic good documentation practices. But for your pivotal studies, you need to adhere to GLP practices, which involves prospective protocols, amendments, review and storage of all raw data, a lot more safety evaluations than is typically performed in animal studies. And because of this, typically, as you all know, contract research labs are used rather than universities. Okay, so as we know, the IND submission, that's really many of us, that's the goal that we're working towards, so that we can finally see this product enter humans and see if it's going to work. And that IND submission boils down to the FDA evaluating whether the potential benefits outweigh the potential risks. That's really what all of this work is for. And we need to explicitly explain this to the FDA. We do not want them to draw their own conclusions lest they come up with a different conclusion than yours. So it's very, very important in that IND that this argument is presented very clearly and cleanly. So I spent 10 years at a small cell therapy company. And as the head scientist, I spent a tremendous amount of time having discussions around fundraising with investors. So I know a number of you will be making that leap from grants into institutional investors or VCs. So I wanted to share a couple of things that I learned over that decade with you today. So the first is that usually they're funding the team more than the idea. So ideas change, landscapes change. At the end of the day, they're really investing in you, and it's really important for you to explain how you'll deal with adversity and work with your team to work to move this product forward. Having some experience is a plus. You don't want to be completely green and expect that they'll hand you many millions of dollars. But in some ways, it is also a minus. They want people that have a fresh perspective and a fresh view. And frankly, the field of biotech is changing in many ways. Diversity, equity, and inclusion is one of those things. And in some ways, if you're not open to having a diverse team, you never will be open to it. And so that's the kind of thing that investors are going to look at out of the gate, because there's an understanding that takes many different kinds of people to make a successful business. And to that point, you want to make sure you do have a lot of different types of people in your startup group. You want to have people of different generations, people of different genders, also different expertise. To just have 3 scientists together probably doesn't give the right level of diversity from an expertise arena that will encourage the investors that you guys can handle and judiciously use the millions of dollars that they're about to hand to you. I think it's also really important to appropriately assign the roles. There's always the desire to be the CEO or, to have that fancy title, but they'll see through that pretty quickly. You want to be assigning titles to people that really can do the job, have done it before, or show that they can grow into that role. And the last thing I would say is I think it's really important to understand the involvement of your investors. Some investors, they want to just write a check and walk away. Those are great investors. Other investors, they want to have a weekly call with you. And not only do they want to hear about the results of study 13, but they also want to be able to tell you how to design study 14. There's pros and cons to that. Certainly for younger entrepreneurs, I think there's a lot of value, but it can also just be a tremendous time sink to manage all of those investors. So understanding their interest and level of involvement up front, I think, is really important. Okay, so the last thing I wanted to do today is talk a little bit about my experiences with academia versus industry. A number of you are working in academia. Maybe you're thinking about starting a company or having your idea be licensed into a company. And there are some pros and there are some cons to coming into industry. So a major pro of working in industry is being exposed to other aspects of product development. There's so many things we just talked about that are not the science, and it's really interesting intellectually to explore those and understand the interplay of all of these different factors in product development. It also tends to put you closer to the patient experience. Not always. Some academic pursuits also can provide that experience. But for someone like me as the head scientist, I was actually going to all of the injections in our clinical trial. And it was really powerful to get to meet the patients that were receiving the treatment that I helped develop. There's also a higher level of fluidity to move between companies, between products, and between science. In academia, sometimes you might work on the same thing for 30 years. In industry, it's okay to move around. It's encouraged. That cross-pollination is valuable, brings new perspectives to the table. And there's also a tremendous amount of teamwork and collaboration. Some people love this. Some people less love this. There's, of course, collaboration and teamwork in academia as well, but it is absolutely critical part of industry success. I will say that a false pro of industry is that you will be mega rich. The acquisitions and IPOs are few and far between, and they are out of your control. And so I think that if you're driven by being rich, maybe go be a banker, because making that your goal in the biotech space is likely to leave you dissatisfied. So there are some things that I wanted to articulate. These are just my perspectives. You know, I would say in industry, one could argue there's less intellectual stimulation. You know, you're not going to talks and conferences as much and reading papers as much. It's not universally true, but I would say broadly, it is a less stimulating environment than being in academia. There's paperwork and there's meetings and there's emails and there's meetings and there's emails. Again, something that's in academia. My hunch based on my interactions with my academic friends is the burden's a little bit higher when you cross over. The other thing that I see is decision-making paralysis. You end up having a lot of cooks in the kitchen. And so sometimes it can be hard to move forward, as opposed to when you're a PI, you can just make the decision and move. Here you have to get buy-in from lots of different department heads before you can make a pretty significant change. There's also a challenge around turnover and hiring. You can have people come and go, and that can be difficult from a training perspective. And you also are faced with loss of control. And as scientists, that can be really hard for us. That being said, at the same time, if you're witnessing that people who are taking over your research should not be taking over your research, for example, business people are trying to lead the science, that is also your cue to move on to the next organization. Because at the end of the day, there's no time for bad science. And there's lots of other companies that would be happy to employ you. But to that point, there's a level of, there's a lack of ownership of your research, very different than academia. You know, if you can move from company to company, you leave your research behind. And for me, that was challenging as I faced leaving a company after a decade, you know, having to recognize that I had to leave that behind and sort of start with new research. That can be taxing for sure. And then finally, there's a lot of variability in the approach to DE&I issues in industry. That really comes from the top, the board and the executive team. It is their organization, and it is at their discretion to define how important and how much they value diversity, equity, and inclusion. And typically that is a fairly well-established approach in each organization. And so it's really important to evaluate that as you consider joining a company, because it needs to be much smaller. You know, there's a small company won't have a giant HR department. It's really on you to understand what is the approach that the board and the executive team has to diversity, equity, and inclusion. And some of that can be seen simply in the composition of those boards and that executive team, but also in the way that they present themselves. You know, at the end of the day, these companies are made up of a lot of different people, and it all comes from the top in terms of how much they want to listen to diverse groups of people. So that is an important thing to either establish ahead of time if you're starting a company, or to understand before you agree to join a company. But at the end of the day, you know, we're all here because we want to get these novel products to patients in need. And there are many, many different ways to contribute to this effort, with basic research, trying to do translational work, as we've been describing here, supporting the clinical development, and then finally the commercial development. And there's just a huge need to get technologies out of academia and on this developmental pathway. And it is a very difficult, expensive, complicated pathway. And programs such as this, they accelerate and enhance these development efforts. And so I wanted to say congratulations to all the groups that presented over the last 2 days on the tremendous progress that's being made on your novel ideas. So congratulations, everyone. And I'd be happy to take any questions now. Thank you.